joining us for Science Cafe, our first Science Cafe of the semester. And we're really excited to be getting up again and giving these science talks. I'm very much looking forward to our first talk to jump off the series with our iGEM team. It's I guess you guys can introduce yourselves, but it's an international genetically engineered machine. There we go. I almost blinked on that. Team at the University of Illinois. It's a really exciting group of undergrads coming together to create these organic machines to provide these services or different processes. So I guess I'll let you guys take it away, and I'll let you go from there. Thank you so much.
Yeah, hi, I'm Vishnu, and I'm one member of the iGEM team. Yeah, I'm a sophomore in physics.
Hi, I'm Megan, and I'm another member of the team, and I am a junior in bioengineering.
Hello, I'm Melissa, and I'm another member, and I'm a junior in chemical engineering.
Hello, my name's Angela, and I am a junior in biochemistry.
Hello, I'm Adrian, and I'm also a junior, and I'm majoring in chemical engineering.
All right, with that, I guess we can get started if there's nothing else. Right. So our project is called Curli Capture, and our aim is to engineer bacterial biofilms to reduce the amount of nitrate pollution coming from fertilizer runoff. So who are we? This is the 2022 iGEM team, as you can see here. Two of our members weren't able to make it to the meeting, apparently, Lauren and Ira, but yeah, that's us. And we are competing in the 2022 version of the International Genetically Engineered Machine Competition, which is a competition that's been going on for 20 years and is one of the premier synthetic biological competitions in the world. So the International Genetically Engineered Machine, or iGEM, competition is based from the iGEM Foundation, which is an independent nonprofit organization dedicated to the advancement of synthetic biology, education, and competition, and the development of an open, collaborative, and cooperative community. So what is synthetic biology in the first place? So synthetic biology is a field of science that aims to create and or modify biological systems that are already present in nature. The founders of synthetic biology are two scientists named Michael Elowitz and Timothy Gardner. So in the 1960s, two scientists, Jack and Monod, created a paper in which they described the process of genetic regulation from lac operon and theorized that you could assemble systems based on that regulation that were completely novel and could be used to do various tasks. So in 2000, two papers were actually published that actually had these synthetic biological circuits. The first was Michael Elowitz's paper, in which he created a biological oscillator or a clock, which he called a repressolator. And the second was Mr. Gardner's paper, in which he created a genetic toggle switch.
So for our iGEM project this year, we want to use synthetic biology to address the issue of nitrate runoff, like Vishnu mentioned. So just a little bit of background on this issue. The global population has been increasing rapidly and is projected to reach 8 billion by November 2022. So because of this, global cereal crop production has doubled in the last 40 years. And to address this issue and meet the increased food demand, farmers have been using more nitrate fertilizer to increase their crop yields. And the reason why we're focused on nitrate specifically is because nitrate is a vital nutrient for plant growth, but it's the most limiting nutrient in the soil. And because of this, this leads to a phenomenon called nitrate runoff, which is the loss of nitrate nitrogen from agricultural systems. And this is a problem because it can get into the waterways and lead to unsafe drinking water and a phenomenon called algal bloom, which is overstimulated plant growth, which can disrupt the aquatic ecosystem. So a real world example of this is in Lake Decatur in Illinois. And because of nitrate runoff, the nitrate N concentrations in the water have been deemed unsafe and past the maximum contaminant level that is present or that is considered safe for clean drinking water. So you can see from the graph on the left that between 1965 and 2010, most of the years have had this unsafe nitrate N concentration.
And so the fertilizer industry is a big part of this problem of nitrogen runoff. And so basically every year, a lot of farmers use high concentrations of nitrate to fertilize their crops because in their opinion, over fertilizing is better than not having enough fertilizer. So even if they have been adding enough nitrate, usually farmers tend to over fertilize with nitrate. However, in reality, only 20% of the nitrate really ends up being used. We hear a lot of like people say, oh, like 95% of the nitrate actually ends up being taken up by the plant, but that is not true. There was an experiment done and only around 20% is actually used. And so since it lingers around in the soil, it actually lowers the pH of soil, which is not ideal for plants at all. And the nitrate in there in the soil also kills off organic matter that plants use to grow better. And it also stimulates microbes to burn off carbon, and carbon is also needed for plants to grow well. And so in general, adding fertilizer actually also stops natural nitrogen production. And so if there is ever if someone ever does not add enough nitrogen, the plant itself can't naturally produce it. And so that's also not good. And so since so much nitrogen isn't being used and farmers are over fertilizing their plants, that's why there's such a big problem with water runoff into waterways, especially for like surrounding cities, agricultural cities. Okay, so in reality, there's actually better ways to grow crops that don't use fertilizers as harmful as nitrogen, such as liming. And liming has actually been proven to make the plants grow a lot better. It softens the soil and increases the pores of the soil, or like inside soil, and it allows water and roots to move around better in the soil, which allows them to grow much better. And so farmers would actually have a lot better yield if they just use liming instead of nitrogen. However, the reason why farmers don't just lime is because farmers actually don't really know that liming works so much better than nitrogen, because a lot of these fertilizer industry companies, they buy anhydrous ammonia, which is a lot more expensive than limestone. And because it's so much more expensive, it's more profitable to just sell nitrogen. And so this is kind of like a huge problem in the fertilizer industry. And so what we decided is since the usage of nitrate in fertilizers probably isn't going to go away anytime soon, we came up with our own solution to this problem.
Lake Decatur supplies 39.4 million gallons of water a day to 87,000 people in Decatur and surrounding communities. However, the water supplied from Lake Decatur has been deemed unsafe to drink due to high concentrations of nitrate. Nitrate is an essential nutrient for healthy plant growth. High levels of Decatur result from nitrate leaching, the loss of nitrogen in agricultural systems. In recent years, there has been an increased demand for nitrate-containing fertilizers. Though this has helped boost crop production, the excess nitrate in the soil has created negative environmental impacts. In addition to creating unsafe drinking water, nitrate leaching can disrupt aquatic ecosystems by causing excessive plant growth. To combat this issue, our project aims to create a novel nitrate capturing system by fusing a nitrate-binding protein, NRTA, to Curli fibers, a self-assembling extracellular protein produced by E. coli. This fusion will be completed by using this pretext bycatcher method. Additionally, we want to attach a nitrate sensitive promoter, so our system is only expressed in the presence of nitrate. To create our nitrate capturing system, we will co-transform 3 plasmids into E. coli strain MC4-100. The first plasmid will be a spy tag, a short amino acid sequence, fused to the main curly protein subunit, CSGA protein. Second plasmid will contain the spy catcher, a small peptide, fused to the nitrate-like NRTA. The spy tag and spy catcher come into contact with each other. An irreversible covalent bond is formed, securely attaching NRTA onto the curly biofilm. So yeah, that's our quick project promotional video, and then back to the presentation.
So our solution is called Curli Capture, and aside from the promotional video, our overall goals are to fuse a nitrate binding protein to the curly biofilm produced by E. coli to create a novel nitrate capturing system. And we want to attach a nitrate sensitive promoter, so this system is only expressed in the presence of nitrate. And hopefully we want to develop a recycling method to repurpose the captured nitrate. So what are curly fibers? Some E. coli strains produce substances called biofilms, and curly proteins are the main components of these biofilms. And the protein monomer CSGA is one of the major subunits of these curly proteins. So when they're secreted outside of the cell, the CSGA fibers will self-assemble into a fiber mesh structure. And overall, we want to use this free-floating CSGA scaffold to immobilize the NRTA protein.
Okay, so what is NRTA? NRTA is a paraplasmic protein that's part of the inert ABCD nitrate transport system that's native in most cyanobacteria. And so the protein itself has 2 distinct domains that kind of form this cleft shape in the middle of its structure. And then this cleft is used to bind nitrate, and it has a very specific binding affinity for nitrate. And so what we want to do is to utilize this nitrate binding ability to capture nitrate. And so how we're doing this is that we are using this spy catcher system. And so as this name suggests, the 2 main parts of the system are the spy catcher and the spy tag proteins. And the larger spy catcher protein and the smaller spy tag, when they come into contact with each other, they will form this irreversible bond. And so we're going to use this to create a CSGA and NRA fusion. And so how are we planning to implement the system specifically? What we're first doing is we're knocking out the native CSG protein in E. coli and replacing it with our spy tag CSGA fusion protein. And what we're hoping that does is that we can utilize the native cellular curly machinery in E. coli to express this fusion protein onto the extracellular matrix. And then that will bind to the NRA, which is also bound to the spy catcher protein. And so the spy tag and the spy catcher will form that irresistible bond with each other. Yeah, and that's all we have. Thank you so much.
Awesome. Thank you so much. That was super cool. If anyone has any questions, feel free to either unmute or you can throw them in the chat. Or I have a couple that I was just going to ask you guys just off the top of my head going through this. One other thing you guys mentioned, the CSGA is coming from E. coli, is that right? Where are you getting the NRT from? Is that coming from E. coli as well, or is there a different species that comes from?
So the NRTA, it's native to like different species of cyanobacteria, and we don't have specific strains of those. So what we did was that we kind of just sequenced it through like IDT, I believe, with the spike catcher in one whole thing.
Very cool. And so you're gonna make this the combination, the CRT, the fusion protein, and you said it self-assembles. What do you think? What is that gonna look like, or how are you gonna get that into the water supply in a way that can bind to the nitrate? Do you have an idea for that?
So basically we're hoping that our final product will be kind of like like a mesh of the protein, like the CSGA protein, with the NERT-A kind of immobilized onto it. So we're hoping to put that product itself probably near waterways or identify areas where nitrate runoff is a bigger issue. And as of now, I think in one of the slides, there's kind of like a diagram. If you want to go to that, Vishnu, it might be slide 5, maybe. Oh yeah, it was that one. Sorry. Yeah, so like right, the one right before number 12. Sorry about that. Yeah, so like there's like kind of like a CSGA mesh, and then those like green dots would kind of be like the bound nitrate. So that's kind of what we're planning, like our product to put into the environment.
Very cool.
I had a quick question on that. So for the, so you're using the E. coli to express the proteins. Are you going to be isolating the proteins separately, or will they actually be expressed on the E. coli? And are you planning on putting the E. coli into the system, or just the protein expression system afterwards?
You go.
Thank you. Yeah, so we are planning to express it in the bacteria first, because we also have a nitrate sensitive promoter. So we're going to express it in the bacteria and see if it can capture it. And then we're going to try to use another strain with the nitrate sensitive promoter to see if we can make the entire system responsive to nitrate. So yeah.
Are you going to try to isolate the protein from the bacteria, or will the bacteria have to express this system, and will it have to stay attached to the bacteria?
I think it'll have to stay attached to the bacteria for the nitrate sensitive promoter. We might try isolating the protein in to just see if it works in the first place, but that's still something we're deciding on.
If the bacteria are going to create biofilms, to kind of capture all of this stuff in nature, to capture all the nitrates, is there a plan then to collect those potential biofilms after it may have, isolated all of the nitrates?
I think so. I think we're going to try to use that to see as like a proof of concept if our system works in the first place.
Great.
Very cool. Well, if there's no other questions, thank you guys again so much for coming and presenting your work. It's very exciting, interesting. I love seeing the concepts that the iGEM team comes up with every year. And this is very exciting work.
Hey, Danny, if you have time still, I have a real quick question for the team. I had an advanced question about biofilms. No, I'm joking. I don't have a PhD. I'm the only one here who does it. So I wanted to know a little bit more about the iGEM experience. How did everyone feel about being a part of the team and how it went this year? How were the advisors? How was the whole process? I'd love to get a little feedback just from everyone very quickly about how you felt about it.
Well, yeah, I can start because personally, I felt that it was a really good experience, and like everyone on the team we work pretty well together, and we have good communication within the team itself. So like while everyone kind of does their own thing, we all know what we're doing. And as for like our advisors, they're also super helpful. We do have like weekly meetings where we can like go over where in the experiments we are, and like do a little bit of troubleshoots and shooting, and then they're always able to like kind of help us with that, and like give us directionality for our project, as well as if we have specific problems in the lab, our advisors also have been like actually helping us in the lab as well. So it's just generally like, for me personally, a very, very great experience.
Yeah, to kind of add on top of that, it's been an amazing experience for me as well, because I kind of started off with like no wet lab experience to now I have like a lot of wet lab experience. I might spend a lot of time there too, actually. But yeah, it's been great. I've learned so much, and I still have so much to learn because we're not even done with experiments. It's been very informative, and I feel like it's something that you can't get in a traditional classroom or even a lab course, to just be working on a project and learning from firsthand experience rather than from a textbook or something.
Yeah, kind of building off that. I've been working in a research lab in the past, but I've never actually like had to plan out my own project, like plan out like which materials are needed, like troubleshooting like a project of my own design. So I think like iGEM has been very helpful in like kind of showing like the like what it's like to make your own project, present the results, and like having to troubleshoot. And also, like Angela said, the troubleshooting part specifically has been it's been very nice to have the mentors and RPI to help us in the lab, since they have much more experience than us.
For me, I really like this experience as well. I'm actually more of a chem major, so I never got to work with bio. And this summer, I learned so much about bio. And even if I didn't know that much, the mentors are also there to kind of point me in the right direction. And so were my teammates. And I really liked how, like everyone else said, you can kind of choose your own project, which is you don't really get a chance to do that in real life. And so I really liked how we could kind of go in our own direction, and then people or our mentors would tell us if we were like doing something right or wrong, and then we could just kind of learn on our own as well.
Yeah, similar to Melissa, I don't really have much of a background in biology, but joining iGEM was a really great intro to what synthetic biology was, and getting to apply the things we learned into a lab. And I also really liked how we were able to choose something that we wanted to do for this project. And I also really like how it's not just bio, there's also like a design portion and like learning how to code and like create a website, which is something I was unfamiliar with, but now I'm pretty familiar with. So it was a really good experience.
How did you guys all hear about iGEM and become a part of it? Especially, you know, since some of you guys aren't necessarily like bio majors, how did you get involved with it or hear about it?
I was reading around a lot of like books on synthetic biology, and they all mention iGEM. It's very prominent because the field is still developing. It's very interesting to see like competition so closely related to its field. So because they all mentioned iGEM, once I came to UIUC, I like immediately applied for like the iGEM team. It was like one of the first things I did, and yeah, it's been like that since.
That's really awesome to hear. Thanks. And I don't have a camera on this computer, unfortunately, but I'd be giving you guys a big thumbs up because I think you're doing really interesting work. And I'm glad I was able to check it out tonight for a little bit. Sorry I came late.
For me, there was an announcement for my like, or there's a bunch of people added to this one like what is it, this one class, and there was like a bunch of announcements for this one class, and one of the announcements was like join the iGEM team. So that's how I heard about it. And then I told Adrian about it, and we were like, oh, that sounds interesting, and that's how we joined.
I think I want to say I heard about iGEM from the MCB roadmap newsletter that they send out every week. Like it was kind of buried in there, but I was reading through, I'm like, oh, this sounds pretty cool. And I kind of read up a little bit more, applied, and it's been from here.
Well, it's super great to hear that you guys are enjoying it and we're able to connect with this team. And yeah, thank you again so much for presenting your work and taking the time out for that. And good luck at the competition. the Jamboree.
Okay, thank you so much for the invite to talk.
Yeah, thank you so much for letting us present.
All right. Well, everyone have a great day, and thanks for joining us. And yeah, hopefully everyone can join us next month for first Wednesday of October. I'm already jumping through months. I almost said November for our next Science Cafe. But yeah, thank you very much. Have a great day.